Welcome, everyone, to the Science of Cooking public lecture series. Wonderful to see so many people here. Really beautiful. Thank you for coming. I usually like to start this week by asking, what do all of these foods have in common? They're edible. Yes. They're fermented, yes. And even if, you kind of knew this because it's kind of the theme of the week, but I think it's good to just kind of take a step back and look at these and consider just how different they are in flavor. I mean, ranging from vinegar to coffee to kimchi to chocolate to wine, all of these really diverse flavors are basically due to microbial products, right? So they have all been produced with the help of bugs. And with bugs, of course, in this context, we mean things like bacteria, yeast, or mold. So this week is very different from previous weeks, I would like to argue. So those of you who have been here before, we've talked a lot about eggs. Do you agree? You're nodding, yes. We've talked a lot about various ways that you cook an egg, the various things that happen when you cook an egg. And we have cooked eggs, but we have also, in various ways, tampered with proteins. So we have heated them, like here. When we made ceviche when Virgilio was here from Lima, you tamper with proteins by adding acid. When you whip an egg white, which is what we did last week for Emulsions Week, emulsions and mayonnaise, you basically mechanically tamper with the proteins. And when you make pesto, this we haven't talked about a lot, but when you make pesto and the pesto goes brown, you can add acids to stop the browning from happening, and so you're sort of playing with the protein that way. Okay, so far we've messed with proteins. This week is different. The reason it's different is basically this. If you've ever fermented anything, who has? Yeah, wow. Special crowd here. When you even just do something relatively simple, which we often don't even think of as a fermentation reaction, we bake bread. What we really, really want to do is keep the microbes happy. What we really, really don't want to do is mess with them. And the reason is that microbes are basically little bags of enzymes. I think the team over here is going to laugh at me and correct me later, but you can kind of think of them like this. And they take various things and they go through a very complicated process, and then they put things out. And we often don't even know exactly what goes on here, or if we do, it's very complicated. But what we care about is what comes in and what comes out. And enzymes, of course, are proteins. So you really, really don't want to mess with the proteins, because then microbes can't cook the food. So sometimes the way I think about this is, so far this course has kind of been about death, because if we took you and we just cooked you, that would not be good. But this is now about life. So this part of the course is about life. The basic way in which this happens, and I think a lot of you know this, is that when you start off a fermentation reaction, there will be some microbes already present, or maybe you add some, the way you do when you bake bread. And these microbes then eat whatever food is there. Maybe it's sugar, maybe it's proteins. They then produce various molecules, maybe ethanol, flavor molecules, all kinds of things. And some of these things, number one, can be intoxifying, right? Ethanol. Number two, can be delicious. They're taste and aroma molecules that are very tasty. And number three is they in some way work to kill all the microbes around them. So not only is this course about sort of life, it's also now about war. Yeah. This is a course for non-science concentrators. Okay. So in the context of fermenting ethanol, for example, alcohol really kills microbes. And you know this because when you ever hurt yourself, you use ethanol or some alcohol on it, on your wound, right? And it turns out it kills yeast too. But it doesn't kill them right away. They can kind of tolerate up to whatever, 13, 14, 15, 20%. And then they aren't very happy either. But up until then, they're just like, they just rule the world. So they're very happy. They're winning the war. So the other key reason that microbes are so good at this, that they're so good at cooking our food, is because they're really good at just dividing and multiplying, right? They're a lot better at this than we are. So you start out with one microbe, right? And it produces 2 microbes. And the first one dies, and each one produces 2 more. And they die and produce 2 more. And this goes on and goes on and goes on. What is this called? Does anyone know? This is basically an exponential, yeah. So here is the equation that would do this, great. In this class, we clap for equations. And what this equation says that if you have some time have elapsed, and this is the time per generation, so for us it's, I don't know, 30 years, something like that, then after a certain time t has passed by, you have n number of microbes, or people, or whatever. So if you were to do this, so here's now the test question, right? If this were a test, you would now sit down and write. So E. coli divide every 20 minutes. If the spinach you had at dinner at 6, so about an hour ago, we all had dinner at 6, it had 1 bacteria on it. How many extra do you have in your body now? And how many do you have tomorrow morning? So I'm not going to make you do this because you're here because you're having fun. But if we were to plug this in, you would say that the time elapsed is about an hour, and the time per generation is about 20 minutes. You would have about 8 right now. That's okay. That's okay. Tomorrow, 15 hours later, a lot. 3 kilos tomorrow. That's a lot of bacteria. Do you think this is actually the case? No. Why? I hear lots of things. Your immune system. They die. Why do they die? Acid. Antimicrobials. There's not enough spinach. Good. At some point, they run out of food, right? And this is quite a lot. So they run out of food, which is lucky for us. That's lucky for us. So the key thing to remember here is, number one, what we really want bacteria or microbes to do is divide a lot. But we want the right kind to divide a lot. And we don't want them to divide too much. Okay, so with that, I believe it's now time for me to hand over. So this is a wonderful pleasure to be able to introduce this team. I think I've been bragging about this the entire semester. Those of you who have been here can kind of attest to that. Sister Noella from the Abbey of Regina Laudis in Bethlehem, Connecticut, is a microbiologist and artisanal cheesemaker. She is joined by Matteo Keeler. He is the co-founder and manager of Jasper Hill Farms and Caves. And with us today here too is Ben Wolf, who is an assistant professor at Tufts University and who actually runs a lab studying a lot of these things. So please join me and welcome this wonderful team.
Thank you, Pia. We also, I think, are going to talk about death because we're talking about decomposition when we look at the microbes. But the subtitle to my talk is Cavemen I Have Known and Loved. Certainly, Matteo and Dr. Ben Wolf are cavemen I do love. So I was very happy that they joined me tonight in presenting. I want you to know that these cheeses were contributed tonight by very generous sponsors that perhaps you've eaten all the cheeses on your plate. But what you ate was Winnemere and Bailey Hayes and Blue. That is from Matteo Keeler's farm in Greensboro, Vermont. The Cowgirl Creamery, located in California, they sent us Red Hawk and Mount Tam. And Whole Foods just sent us so many cheeses for this. And it was a Kalbeck Gruyere, that was the hardest cheese you had, and the Taleggio, which was a smear cheese with the orange rind. And then you had some Bethlehem cheese from the Abbey of Regina Laudis. So tonight we want to talk a little bit about the origins of cheese making and microbial ecology involved in cheese making. And give you more of an idea of what happens when someone makes cheese. Have many of you made cheese? Many people here have made cheese. Oh, that's great. Well, they say that 1 gram of soil contains 10 billion microorganisms and perhaps thousands of different species. So what we do is put a cheese in a cave so it can be close to the soil. So I want to show you, this is a cheese from our Abbey. This is a Bethlehem cheese that's come out of the cheese press. We've washed off the salt. Were you to eat this now, it would not be tasty and it would be rubbery. You might taste a little bit of yeast, but perhaps not. But what's in here at this point are lactic acid bacteria. And they're consuming the lactose, the milk sugar, turning that into lactic acid. What happens in the cave is a transformation. This is about day 4 of the ripening process, and we are going to look at these microorganisms up close and personal under an electron microscope. This is a natural process. We never added commercial cultures of lactic acid bacteria, as most cheesemakers do. We did not add fungi. We'll find out more about this microorganism. This is called Geotricum condidum. It makes this white powder, and I did my doctorate on Geotricum condidum, so it's very close to my heart. And then here we have a cheese. This is finally ready to be eaten. You ate this cheese. But this is a very complex flora that has now grown. So there's a succession of microorganisms on this rind, but we never added cultures of fungi or bacteria. So it's all natural. So Cambert, who was a writer, said that cheese is one of the primary symbols of mankind's passage into civilization. So I want you to come down this passage with us tonight. I think it's helpful to look at the etymology of the word cheese to understand a little bit in what happens in the process, which is the Greeks. They called the basket that they used to drain the whey out of the curd the formos. The Italians then call that the forma, and that's where we get formaggio and fromage. This is the Proto-Indo-European root of the word, which is quat, which means to ferment or become sour. And from that, we get the Latin caesius and the Western German caseus, from which we get the word cheese. So what you have to think of is liquid milk is becoming solid, and it's also through an action of fermentation, becoming a fermented food that is portable and delicious. When did this begin? Well, we're not sure, but this here is the oldest milking scene that has ever been found. This is in the Libyan Sahara and taken about 6,000 BC, and we have to take a closer look. Don't think of the Sahara as the desert as you would now. It was a more temperate climate. So here we have these cows, and there's a figure. It's a black figure in the middle there. You see his back? They say that he is actually milking. He's milking into a white container, and behind the cow is a calf, and she is there to help the cow let down her milk, because you can't milk some of these cows without having a calf nearby them. These cows don't have very big udders, so they probably didn't give a lot of milk. What we see on the left is what they think is an image of a corral. And to the right of that are people making dairy products. So how did I get into this cheese business? Well, I have to introduce you to another caveman. He's called St. Benedict. And he is the father of Western monasticism. He was born in about 480. And his parents sent him off to Rome, which was common. He was born in Nursia, which, sad to say, that was where the most recent earthquake in Italy took place. So the basilica over his birthplace was destroyed. So he went to Rome to study, probably 18 years old, and at that point it was really the decline of the Roman Empire. And so it was really run by barbarian chieftains, and I guess he didn't like what he found. So he left, and he went off to live in a cave. He lived in a cave for 3 years. And at that point people came to him for advice, and he became inspired to start monastic communities of men living together. So the motto of the Benedictine is pray and work. And we do plenty of both. And he says in his rule, when they live by the labor of their hands, then they are truly monks. Now, the ecologist and microbiologist René Dubos said, the Benedictine monk was the first scholar to get dirt under his fingernails. So that is what we do at our abbey. We try to have a self-sustaining farm, and we do plenty of work and get our fingernails dirty. The Benedictines also developed European technology and science. And he continues, René Dubos, for the first time in the history of human institutions, the Benedictine Abbey created a way of life in which the practical and the theoretical skills could be embodied in the same person. What do I do in terms of work? Well, for me, it began with this cow named Sheba. That's me as a novice, feeding Sheba. We have since switched. You see, she's a Holstein, but now we have what are our Dutch belts, who give richer milk. But I was asked, I milked this cow Sheba, we had milk, and I was asked to try my hand at cheese making. I tried making it, but I went to our abbess and said, you can't learn it from a book, it's not working, and prayed for an old French woman to come. Instead, a young French woman came. And she taught me the recipe for traditional Saint-Nectaire. Her grandmother taught her how to make this cheese. It's from the Auvergne, France, been made for centuries. We will get to the Auvergne later on our trip tonight. But first, my community decided to send some of us off for advanced degrees at UConn. And I ended up in molecular and cell biology, in microbiology. My major advisor from UConn is here tonight. And he's wearing a rubber nose in case I embarrass him. But what we wanted to see is, could we see what happened on this rind naturally, since we did not add any cultures of bacteria or fungi? Could we visualize that on the cheese rind in situ? Because as dairy microbiologists in the past, what we would have to do is isolate each microorganism, get a pure culture, then analyze it for genetic or biochemical diversity. And so we wanted to see, could we see what was happening on the rind itself? So we studied the rind using light microscopy and electron microscopy. So what we have here is the techniques that we used. And that's the Bethlehem cheese on the left, that young one, and then the aged one, and these are the techniques. So here is Bethlehem cheese at day 0. Under the electron microscope, this is what it looks like. It sort of looks like the surface of the moon. It's smooth. And that under the light microscope, that's what that looks like. So that's the surface of the cheese. The arrows are showing you there's nothing growing on the surface. What we see are isolated pockets of lactic acid bacteria within the curd. But by day 2, something very dramatic has happened. So are you ready to see that? Same magnification. So there's something embedded in the rind. And we can see in the top also, there's something growing there. Basically, what those are budding yeasts. Those are yeast cells that have grown into the rind. And then by day 7, there's a great change. This is, again, the same magnification. So this, I want you to just remember, this is what that would look like day 7. And what we see is Geotricum candidum, and then there's these hairs that are growing on the cheese. You can look closer later on. And that's a type of fungus, and we call that mucor. It's a zygomycete. So every hair on that cheese has a ball on top that's called a sporangium that has thousands of spores on it, and they break off. And this is now what it looks like with the light microscope from the top going down. And there are little round balls on the top of the surface, and that's the hairs. So mucor is fine on a cheese like this. It's fine on a tome de Savoie, but it can be a major problem if you're making a reblochon, or cheese that you want to look white and orange, like a smear cheese, especially terrible on Camembert Brie. And once it invades a cave, it's very, very hard to get rid of it. One way they say to help it is to get a good implantation of Geotricum on that type of cheese. and that can help inhibit mucor. But in some cheeses, it's great. On others, it's a real contaminant because you think thousands of spores on that one hair, that ball. And then what we call this next one just, it's a dry cheese now. And what we see here, though, underneath, and it's hard to see here, is a pink surface, and this is Trichothecium roseum. And that there are thick hyphae that we see there, interdispersed with the hyphae of mucor and the spores. Here is the pink powder. That's Trichothecium roseum. It's two-celled conidia. And my French cheese master was so surprised. She worked with me for 2 days, went back to France, and came back for two years later and was very surprised that we had the same fungi on our cheese in Connecticut as she did in the Auvergne. And she didn't bring them over in her pocket or in vials. They grew naturally. And when she saw this pink powder, she said, "You have the flower of the molds." And so this is one that they love to have on their cheese. It's called Trichothecium roseum. It's very proteolytic. It breaks down protein. So in general, when we talk about decomposition, we're breaking down the milk sugar, lactose, the lipids, the fat, and the protein, proteolysis. And that's what gives cheese its flavor and the consistency changing so dramatically during the ripening process. So this is basically, you're looking at this cheese right now. And we call this fungal debris. There it is. Speaking of death, that's kind of what's a lot of dead cells here and a wonderful cheese. And underneath that, or much higher magnification, is what we call, it might be Brevibacterium. It's a group of bacteria, the coraniforms, and they kind of look like clubs. And I just wanted to show you, this is Winnemere. This is Matteo's cheese from Jasper Hill. And Brevibacterium is the orange bacteria that you would see like on a true Munster, and or many of the monastic cheeses, or those smear cheeses. Some of the smelliest cheeses is because of Brevibacterium linens. Brevibacterium is sensitive to low pH, so it grows fairly late in the ripening process because the lactic acid bacteria, they first acidify the curd, and then we have the yeasts that consume the lactic acid and start bringing the pH up, less acid. And then we have other fungi that can grow in acidic conditions but also raise the pH. And that's when you can have something like Brevibacterium is now finding a home that was created by the other microorganisms. So it's microbial ecology. There's this finely tuned ecology going on the rind, and we did not add these microorganisms. They grew naturally. So basically, what we saw in our study, we discovered really that the hyphae, when you look at this, these are aerial hyphae that are growing up, these hairs. But then something like Geotrichum has vegetative hyphae that grow down into the cheese surface and almost till the surface and aerate the surface so other microorganisms can grow down into those channels. That's one of the things that we found. We were surprised to see that was one of the perhaps roles of these microorganisms, is that tilling. So because we had not added any commercial strains, we wanted to be able to have access to other fabrications that were not using commercial strains. So luckily, I got a Fulbright to France and could go to France, and I had the host lab in Pouligny at the Institut National de la Recherche Agronomique, and I traveled 30,000 kilometers to get samples of native strains, and we decided to concentrate on Geotrichum candidum. We actually are going to pass around some petri dishes of Geotrichum, which Ben Wolf's lab at Tufts prepared for us. It's described as a yeast-like fungus. So what I wanted you to see in these dishes, and it's quite odoriferous, is there are two different colonies there, and one is going to be more yeast-like, more furry, and one's going to be, more fungal-like and furry. The other will be creamier, and that is yeast-like. That's important for the cheesemaker because sometimes you want a strain that's more fungal-like and thick and fluffy, or like velvet, but sometimes you don't want it too thick. But if you have a strain that's too yeast-like, when you go to turn the cheese, it can pull off pieces of the rind. So the cheesemakers get to choose commercial strains that are more fungal-like, yeast-like, or in between. And it's ubiquitous. It's on Brie, Camembert, Tome de Savoie, Saint-Nectaire, so many of the chèvres. And it's a very important microorganism in terms of the breakdown of protein and fat. And this is showing you the two forms of it, filamentous and then what we call the arthrospores that form through this disarticulation, so almost like arthritis, this joint that breaks open produces those arthrospores. So first we're going to go to the Haute Savoie, and here I think he's such a good example, Frère Nathaniel, at the Abbey of Notre-Dame de Tamier, which is in the Haute Savoie. This is a Cistercian abbey, and it's at 900 meters altitude. The monks first came here in 1132, and they started agriculture and cheese making. Probably it was a cheese like Gruyere or Abondance, a tome. And they really started cheese making not only to support their community, but to feed the pilgrims that were passing through the Alps. But they had to leave the abbey during the French Revolution. And when they came back after 1860, they came back with a recipe for a Trappist cheese, a lot like a Saint Paulin. And from that, then later they developed this cheese, which is Tamier, which is fairly close to a Roublechon from that region. So this man here is Frère Nathaniel, and he went to dairy school and worked for 9 years in the industry, and then he became a Cistercian monk. And I think the things that he has put in place for their cheese has been really brilliant, and sort of, he's a pioneer, I think, in some of the things that are going on now in this country, working with cheese and cultures. You'll hear more about that later. First of all, this is an AOC cheese. Appellation d'origine contrôlée, or protégée, means that this product came from this land, and in the past you had to use a certain breed. In the Haute Savoie, it's the Tarentaise and the Abondance. You're a little shy at first. I think the Tarentaise, she's the brown one here, is one of the most beautiful breeds, native to the Haute Savoie. And then the Abondance is the cow with the white face. And they're pretty nosy. So what Frère Nathaniel did, and should try to encourage a sense of this cheese being of his earth and that region, because the monks no longer milk cows themselves. He uses the milk from 11 local farms who only are fed from local pastures and local hay. And that is part of the legislation for an AOC cheese. The feed has to be from that region. So what he did is he isolated lactic acid bacteria and other bacteria from local hay and milk and the cows, and worked with a culture company to make cultures that he could then add back to his cheese.
Brother Nathaniel has developed over the years a machine that would be used to fill the cheese molds. And he worked at this for many years until it was exactly what he wanted. Because he knows what he wants in terms of the quality of his cheese on a small scale, and he's so familiar with the technique, he managed to scale up the process and not lose the soul of the cheese.
So you can see that that's the beautiful cellar that they have, the cave. It's actually under the choir, and that's what their cheese Tamier would look like. So I spent a lot of time in that cellar with him, and we talked about many things, and I asked him some questions. The woman who produced the movie, these are clips from her movie, decided she wanted me to ask questions in French and then translate his answer. So you'll have to put up with my French here, but I think Frère Nathaniel has important things to say about the relationship between monasticism and being a good cheesemaker.
Et aussi, vous êtes une moine?
Oui.
Oui. Et est-ce que vous pouvez parler un peu de ça? Parce que je me souviens, vous avez parlé un peu du rapport entre votre vie monastique et d'être une fromagère.
La vie monastique, elle est bien connue, surtout la vie bénédictine. Et, prière et travail, ce sont deux poumons qui nous font vivre.
Work and prayer.
Voilà. Les deux sont, bien sûr, on peut bien sûr prier en travaillant.
Yes, we can pray while we work.
Et je crois que être artisan, c'est-à-dire travailler de ses mains, est vraiment une participation à l'œuvre créatrice. Et lorsqu'on en prend vraiment conscience, c'est extraordinaire. D'autant plus que le travail du lait, c'est un univers extraordinaire, complexe mais extraordinaire.
So you're saying that to work with our hands as an artisan is to participate in creation.
Tout à fait.
Okay. And the world of milk
Voilà.
is an extraordinary world.
D'autant plus qu'aussi, travaillant avec des producteurs de lait, on est amené à être en relation avec un monde du travail, un monde agricole, et aussi de vivre avec eux le rythme des saisons.
So when you work with the farmers, you're in touch with the life of agriculture and with the seasons, et aussi peut-être les méthodes traditionnelles. C'est important pour vous?
C'est la condition essentielle, à mon avis, pour produire un lait qui permette de fabriquer un fromage au lait cru. À savoir qu'une agriculture dite raisonnée à base d'herbe, à base de foin en hiver et d'herbe en été, simplement on retrouve ce que l'animal doit normalement ingérer. Et on a montré que finalement, grâce à cette méthode de production, on avait moins de problèmes de contamination de bactéries pathogènes parce que la bête vivait tout à fait normalement.
So you're saying the animals live according to the seasons with their food, and they eat hay in winter and grass pasture in the summer, and that it helps to prevent pathogens in the cheese.
Oui.
If the animals live according to a more natural diet.
Parce que la bête vit à son rythme, elle n'est pas poussée, elle n'a pas, donc elle produit elle donne une matière première qui est à la fois proche de ce qu'elle produirait naturellement et en même temps suffisamment chargée de microbes naturels pour pouvoir fabriquer un fromage au lait cru.
So the animal isn't pushed, and also then she produces milk that would have the microorganisms more natural from the region.
Donc vous comprenez que pour moi c'est essentiel qu'il y ait un lien très fort entre la méthode de production du lait et la fabrication du fromage au lait cru.
There's a relationship between the method of production and how the raw milk cheese is made.
Et en plus, comme vous disiez tout à l'heure, ça s'inscrit dans la vie monastique, tout simplement, sans difficulté non plus.
Pour vous, c'est une chose monastique, le repos.
C'est une voie. Il y en a d'autres, mais je pense que c'est une voie.
He's saying it's one way of relating the monastic life to making this traditional cheese. And I think also for the monks, these families from whom they buy the cheese, the milk, they are families who actually kept the land going during the revolution when the monks couldn't be there. So there's a real relationship of trust and interdependence between the producers and the monks, and I think he expresses that very beautifully. So now we have to move on to the Auvergne, Massif Central. So my cheese master was from that region. So we were going to go on to Saint-Nectaire, which is this beautiful volcanic region with the Salaire cows. I have to confess it might be my favorite region of France. Also very Celtic roots. This is a dolmen. And this is the mysterious Salaire cow. She's so maternal that you cannot milk her without the calf being tethered to her neck, and it is mass confusion. But they also, I mean, she's a rustic, tough cow of the region. And now many of the farmers are bringing in Montbéliard, who comes from an entirely other region. She comes from Franche-Comté. And they're having trouble with the hooves. Whereas the Salaire, she was made for that region. She comes from that region. But she does present a few problems in terms of milking. So this is a Celtic region, and the tools that you would use to cut the curd, the poussador, the moussador, those are Celtic words, but actually then the French words, like a moussador, what we're doing there on the right is using a paddle to push the curds through the barrel. That means really mettre dur, to make something hard. We're bringing together pieces. That's what that moussador. The poussador is something, an implement you would use to press the cheese. So then you're pushing something to make it hard. Pousser dur. So I want to bring you to the caves of Saint Julien. I spent a lot of time there, and they are absolutely beautiful. But it's kind of a sad case also of natural history, in the sense that in about 1890 to 1900, we had the terrible outbreak in France of phylloxera, which destroyed the vines of France. These caves used to be wine caves, but the Auvergne never recovered from that. So they became more a center for tires like Michelin in Clermont-Ferrand. This man you're going to hear, Alphonse Ballant, his family has been making Saint-Nectaire for 8 generations, and he has amazing caves here. He also has troglodyte caves that people lived in on his land. And so his grandmother bought these caves, and now they're used for cheese. And they were carved in volcanic rock with pickaxes. Volcanic rock is wonderful for cheese because it holds moisture, and we need high moisture, high humidity in a cheese cave. When I first met Alphonse, he said to me, and what you're looking at actually are thousands of young Saint-Nectaire lying on rye straw in this cave. And he said, the inspectors and the regulators can't kill us. These cheeses are the collective memory of France. So, is he not a caveman I would love? So we have been friends ever since.
Alphonse is a young man who loves his region passionately. His family has made cheese there for many generations. And they use caves that were dug in volcanic rock over 200 years ago. And when I first visited his cave, really, it is the most beautiful cave that I have seen. Because a cheesemaker used a cave that is built into the earth, has been there for centuries. There are strains of fungi that developed, and the cheesemaker saw, if I keep the cave at this temperature, or if I add rye straw, this gives a great cheese. The first goal of my work in France was to collect natural local strains of the fungus called Geotrichum candidum, and then to test the strains I collected, or part of them, for diversity. So how we tested for diversity, we'd use 2 techniques, PCR to amplify DNA. Now, at this point, this isn't necessary, what many microbiologists do now to study biodiversity in terms of genetics. We were the first ones to use this technique on Geotrichum candidum and to be able to differentiate strains. So Geotrichum is the genus, candidum species, and then we want to differentiate different strains within that species. So we use these techniques, amplifying the DNA, and then using this technique called rapid to see if we could even differentiate strains. And at that point, there were no published lists of the sequences of primers needed to differentiate strains, so we just had to keep testing to find ones that would give us results. So what you see here is we finally were able to find primers that worked, and then anything that was less than, I think, does that say 88.3% similarity, we could say were different strains. So here we're looking at strains
I should say isolates, at first you don't know if they're strains, from the Auvergne. So basically what we found was 13 out of 18 isolates were unique strains, which means that's very high diversity. And we could say two, like on the top here, 2 of the strains are only 3.5 kilometers apart, extremely different. On the bottom, 2.6 kilometers apart, extremely different. Now you might think, well, you know, who cares? But we do want diversity because these microorganisms have enzymes. Pia was speaking about that. And basically, their enzymes are breaking down fat and protein. And we are benefiting from the byproducts that they produce. So that's what gives cheese its flavor and consistency. Also, it gives the cheese a certain appearance. Like a Marcellino, they say it has a jolly robe because of the Geotricum strain, and that's important. Also, reducing bitterness. One of our studies was aminopeptidase activity that cleaves bitter amino acids in peptides. And Geotricum is known to reduce bitterness, especially in cheeses that also have penicillium. And they say bitterness is probably the major flavor defect in cheese. And people, I guess. So it also neutralizes the cheese surface, bringing up the pH. And then some strains, there's research going on now because you know about Listeria monocytogenes. You've heard about it in food poisoning outbreaks. And people are interested in competitive studies. There are some strains of Geotricum that can inhibit Listeria. So those kind of competition studies are going on. Also inhibition of aflatoxin. And then some strains inhibit mucor, which is what you wouldn't want on a brie or a camembert. This is kind of summing up our research in France. And the person speaking is actually David Benson, my advisor.
She eventually wound up with a couple of hundred different strains of Geotricum candidum and was able to classify them genetically in terms of their diversity and was able to relate their diversity to the type of cheese that was being produced, where it came from in France, and came up with some really remarkable findings.
We found incredible diversity in the strains that we isolated from farms that could be 10 kilometers apart. And one might say, well, who cares? But we think that from one of the studies that we did, this could contribute to the cheese flavor.
The diversity of the local strains of microorganisms in a region contributed to the diversity of cheeses in France. And just as you want to save a certain kind of tree in the rainforest, you want to save the microbes that are part of a region because they're the ones that have contributed to the flavor and special unique character of a cheese. So you may not think that microorganisms are important, but for instance, for the French, they consider these microorganisms part of their patrimony.
So now we want to bring this back to America so you can see what is happening right now in terms of artisanal cheeses and the scientific research that is going on, the collaboration between Dr. Ben Wolf and Matteo Keeler. And I don't know if we pointed out this book, did we?
I don't think so.
Oh, this book, which is the Oxford Companion to Cheese, just came out this week. Matteo wrote the foreword to the book, and it's quite a beautiful book.
All right, I'm actually going to step out front here. So my name's Matteo Keeler, and I started Jasper Hill with my brother Andy. This is Jasper Hill Farm. I live up here. We make the cheese down here. The barn is right there, and it's attached to the building. We never have to go outside. You know, it's northern Vermont. And it's but we started Jasper Hill really to try and satisfy 3 fundamental needs: meaningful work in a place that we love with people that we love, and that's basically a whole life for us. The meaningful work part is really the conservation of our working land. We have this pastoral beauty that's threatened by global economic forces that are far out of control of the people that live in these rural places. There's 670 residents in Greensboro. And we were looking to demonstrate that you could still make a good living milking 40 cows on a hillside farm in northern Vermont. And the rest is kind of a snowball. To tell you the truth, we're making it up as we go along. So there's my brother Andy started on the farm, on the barn side, milking cows. I started as a cheesemaker, and our wives forsook their careers to join us, and I want to honor the women in our lives because they really are responsible for getting us up over the edge. It was a lot of family labor and a lot of sacrifice that has enabled us to build our business. In 2006, we had something that happened to us. I mean, we were responsible for it in some ways, but we had been ripening this cheese for Cabot Cooperative, Cabot Cloth-Bound Cheddar, and in 2006, it won Best of Show at the American Cheese Society, and we had enough cheese to sell 25 wheels a month to a national market that wanted to strangle us. And we had to make a decision about how we were going to proceed and whether we were going to just let Cabot Cloth-Bound go, or whether we were going to leverage this opportunity to reimagine the way farmstead cheese businesses might operate in our community. And for us, cheesemaking is really about developing place-based products, revealing our terroir. And terroir is a complicated word. It means taste of place, but so much more in French. It's a word that's loaded with social, cultural, economic considerations. And for us, taking a long view, we know that the power of cheese cheese has the power to support entire communities. And you go to places like the Jura in France, where you have one cheese comté that supports thousands of dairy farms and professional activity, and there's the whole world in the Jura revolves around this one cheese, because this one cheese is like an embodiment of their heritage and their landscape. We have appropriated some of these ideas and adapted them in very American ways. We believe we have probably improved them in some ways. Every time I come back from France, I'm like, woo, this is great. I'm so glad I'm not like shackled by tradition. It's a real privilege to be able to invent our traditions and to leapfrog ahead. And I feel so privileged to be making cheese now. I used to describe being a cheesemaker as being like God, except blind and dumb. And what's happening now, and we'll get into this in a bit, is really that somebody turned the lights on and we can see. Still dumb, because we don't necessarily understand what's going on. So Cabot Cloth-Bound won Best of Show, and we took that ribbon to the bank in our contract with Cabot to the bank, and we built a series of underground cellars that would enable us to ripen any kind of cheese a producer might dream up. And the idea here was to lower the barriers to entry for artisan cheesemakers by simplifying the business model. And we continue to evolve that approach in order to try and support farmers and cheesemakers in our community and plug them into a different market that can deliver a return of a different magnitude, order of magnitude. So we blew out the hillside and poured this monolithic concrete structure and then buried it. And so the recipe really goes something like dig big hole, dump in lots of money, bury. Right? And in the fall of 2008, we had cheese to sell, right as the economy was starting to really go right down the toilet. And it's been an awesome time to start a business. It really has, because we started so lean, and I'm going to stop talking about our business and start talking about microbes in a minute. I'm sure some of our customers are out here, and I want to honor cheesemongers and our customers who during that time frame really stepped up to participate in the survival of our business, and we really feel connection. We're at the other end of a long chain, and without our customers, we would not be able to do what we do. So I know you're out there. Thanks. So I'm going to get into some of the nitty-gritty, the making milk and making cheese and aging cheese, but I just wanted to start with this slide. It's just a reminder that in order for there to be milk, there have to be babies. And we live in a world where that isn't necessarily obvious, even though we're all mammals. So we milk 250 cows on 2 farms, and a lot of what we do is manage fertility and estrus cycles in order to begin this biological process of making milk. So what is milk? You know, milk is fairly simple. It's mostly water. There's lactose, there's fat, there's protein, and there's minerals. And all these components play an important role in what comes next. As cheesemakers, what we're really doing is using microbiology to manipulate and transform the chemistry of milk. No 2 milks are created equal, right? Milk is so place-specific. There's the design of facilities that really influence. It's like the built environment has its own microbial ecology, right? Design is a huge part of microbial ecology, and humans are right in the middle of that. So as we start talking about this idea of terroir, it requires humans to reveal that terroir. We've got species, breeds, feeds, microbes, the environment, and the quantity and quality of components. I'm not going to spend too much time talking about feed, other than to just harp on this idea of grass-fed. Everybody wants grass-fed. But a cow, a lactating dairy cow, needs energy. Way more energy than a simple grass diet can provide. A cow that's making 60, 70, 80, 100 pounds of milk every day needs a lot of energy. And a cow cannot get all that energy from just plain grass. So there's a lot of genetic contributors to modern dairy cow, but we do feed a little grain in order to give them the energy required to metabolize the protein that they're getting from their grass. It's not a grain-heavy diet, but we all need a balanced ration. And I just want to tamp down this idea that grass-fed cheese is something that's good. Generally, milk from purely grass-fed cows makes horrible cheese. It does. Because they're ketotic. It's like the Atkins diet, right? And that milk that's full of ketones produces flavors that taste like new shoes, yeah, or Band-Aids, right? And generally unpleasant flavors. So where do microbes come from? Microbes are coming from the feed. So the microbial ecology of raw milk is the sum of the practices on a farm, right? How you bed your cows, how you feed your cows, your milking protocols, how you clean your equipment, how you store your milk, how long you store your milk, the quality of your water, whether your employees wash their hands or not, right? We milk cows, but what we're really doing is farming microbes, and everything that we do on our farms is really using that idea that we have these practices and they're going to create the microbial ecology that is the foundation of flavor potential in our cheese, because there's a linear correlation between microbial diversity and complexity of flavor. Quickly, there's 3 different types of classification. You've got cold-loving bacteria. Generally, cold-loving bacteria, not good for a cheesemaker. But you know what we do? First thing we do when we milk our cows is we cool that milk down, right? And so we're suppressing the growth of all these good microbes and encouraging the growth of the bad ones, right? So generally, we try and process that milk fresh, warm, right out of the cow. Milk was never intended to exist outside a mammal. We saw this from Pia. Yeah, this is what happens. You know, and if you think about cheese making and fermentation as the application of selective pressure, you know, you're going to be favoring different communities depending on what temperature, what humidity, time. All these things are going to favor different groups of microbes. Whether a cow's on pasture, you know, you're going to get more diversity, right? On hay in the barn, less diversity. And you'd think that there'd be a lot more microbes in the barn, right? Kind of cow shit all over the place, and you know. But the fact of the matter is that we're able to control the animal environment much more closely than we can control where a cow is going to, you know, poop and lay down out in the pasture. Right? Plus, there's all kinds of different microbes in the grasses, and most of these microbes are good. They're going to add complexity and flavor potential to cheese. Same milk here. So this is in the farm bulk tank, and this is in the silo at the cheese plant. Right? You can see how that cold temperature is really shifting the relative index, the proportion of the different functional microbial groups over time. And that's cold. Generally, gram-negative bacteria, not so good for cheese making. Get like bitterness, Pseudomonas, fluorescence, listeria, none of the good stuff. This is a picture of our herd of Ayrshires on pasture. And this is a picture of us cropping a few acres of hay. We're possibly the only cheesemaker that's feeding dry hay to at scale that I know of anywhere in the US. There's probably others out there. We're feeding 250 cows dry hay. And we do that in order to shift the microbial ecology of our raw milk that's destined for the production of raw milk cheese. We heard the brother at Tamier talking about working with the natural feeding cycles. What he was really saying is that silage, which is a fermented feed, is a rich microbial environment that has a potential to produce cheese with defects and pathogens. which is why we only feed dry hay. We've been able to manage listeria out of our milk supply by changing the way we feed our cows. So what is cheese made of? It's miraculous, really. You've got the same 4 ingredients and you've got 1,000 iterations, right? How cool is that, right? Basically cultures, rennet, shown as scissors. So we like to think about enzymes really as clipping proteins. Salt and other stuff, we don't really mess with the other stuff too much at Jasper Hill. And then in the cellar, how do we manage what happens, that biochemical cascade that is a ripening cheese? We have a few tools. And if you think about the concept of selective pressure as being the means that an affineur, or a cheese finisher, or ripener uses, a degree Celsius can create a pressure that's going to favor one microbe over another. A couple of degrees of relative humidity are going to favor one group of microbes over another. And what we're doing is really using mechanical action, brushing, managing our environment, temperature and humidity, managing gas pressure, air exchange, essentially looking at, if you exchange the air, introduce more oxygen, you're going to favor molds. If you allow proteolysis to produce ammonia and allow that ammonia to build up in the atmosphere, it's going to exert this gas pressure on the rind. It's alkaline. It's going to favor bacteria and the B. linens and other types of yeast, right? So just by switching on your air exchange, you can radically alter the conditions that are going to favor one group of microbes over another. And ultimately, that's what affinage is. To review, we've got the components in your milk and in your cheese. You've got ripening agents, and really what we're trying to do by adding starters or by managing our herd in such a way is to create these microbial populations that will live and then die. Because when they die, they release their internal enzymes, and those enzymes are fundamental to breaking down fat and protein in order to release the flavor. Long chains of amino acids, proteins, they don't have flavor. We can only really sense them when they're when they're short, and then there's aroma, and there's these volatiles, and we can access what's basically locked up in these proteins. That shredder is really what we're accomplishing when we aggregate proteins in a vat. Rennet begins that process by clipping kappa casein and those trillions of microbes that are growing in that milk, and then dying and releasing those internal enzymes play a huge role in flavor production later on during the ripening period. So affinage is really about realizing the full potential of a cheese. I'm going to hand this next part of the conversation over to Dr. Ben Wolf, who we've been working with. We've had the great pleasure to be working with microbiologists at Harvard for years now. Dr. Rachel Dutton, who was funded for 5 years to use cheese, the rinds of cheese, as a model system to study microbial interactions, fell in love with our cheese at Formaggio Kitchen, and wrote this grant proposal and got funded, and then came, knocked on our door, spent 3 months making cheese with us, and used our facilities, which is like a microbial zoo, to really start this incredibly exciting research. And as a consequence, we've actually built a lab. We can extract, amplify DNA. It costs us $4 now to get a partial sequence. It's crazy, right? But more than anything, it's given us the opportunity to interact with people that are way smarter than us. And Dr. Ben Wolf, who's working at Tufts, teaching at Tufts now, is really spearheading some very exciting work, and he's going to tell you a little bit about how that works.
This will be really fast, and just because I know you're all hungry and you want to hang out with these guys and maybe look in the microscope. So what we're doing is trying to answer more deeply some of the questions about who's there. So you've been hearing a lot about Geotrichum, Brevibacterium, Trichothecium, one of my favorite fungi, Scopulariopsis, which is so fun to say. And so what we're trying to do in the lab is understand not just on a single cheese like Bethlehem or not just like a single cheese like Bailey Hazen Blue, but all cheeses in the world. What microbes are out there and what are the patterns of diversity of those microbes? So what we do is I like to tell people it's food CSI. So just like at a crime scene, you go collect DNA and you go back to the lab and there's sexy lights and good music, which does not happen in the lab. What I wish it did. What we do is we take a cheese sample, a jar of kimchi, a piece of miso, and we extract its DNA, the microbes that are there, and then we amplify up these very specific genes that tell you what the microbes are. So what are the bacteria? What are the fungi? And sometimes even viruses. And you put it on a fancy, really expensive machine that can rapidly sequence tons of DNA at one time. And then at the end of the day, what you get back are these lists of who's there. Just like at a crime scene, who is the criminal? We take our DNA, instead of putting it into a criminal database, we put it into a microbial database to figure out what those microbes are that are on a cheese. And you might ask, why do this? Well, it can help us figure out who's out there, and sometimes when things don't go really in the right direction, you can figure out why, what microbes are driving the cheese in the bad direction, or what's on this amazing cheese in France that maybe we want to try to recreate in the United States. Don't tell the French that, though. This is our rainbow of cheese. So essentially, this is our big data set that we collected from 137 different cheeses in the United States and throughout Europe, and asking what microbes are out there. And so this is, again, DNA sequence data, and it's telling us these different colors that you're seeing are different types of microbes, and this is at the genus level, so like maples, pines, and oaks, but for microbes. And so what we're seeing, this up here is the data for the bacteria, and this down here is the data for fungi, and each of these columns represents a different cheese. And this tree up here, don't worry about that, it's just clustering the data together based on how similar they are. So this is really important because no one actually had done this. So Sister Noella had done this amazing work with Geotrichum and a few cheeses in France, and folks in Italy and a few places in the UK had sampled their favorite cheese. But no one had sat down and just blasted a whole bunch of cheeses together at once. And so you can see there's some really cool patterns. Like you see a lot of these bright colors over here. These are bacteria known as proteobacteria that have tails and can swim around in wet environments. So these cheeses tend to be, they're not tails, they're called flagella, but they're like tails. And so these bacteria love wet environments, so they love really wet cheeses. So for example, some of the wetter wash-drying cheeses that have the stinky foot odors, there's a lot of those bacteria on those. And so you can see some other things. So there's this fungus that I love to say, Scopulariopsis there, which is very dominant on some cheeses like Bailey Hazen Blue and other cheeses. And I just want to point out there's some two stars of the show in this particular data set. So this cheese right here, this column right here, I'm so jittery from so much coffee, it's hard to point it out, but that's Sister Noella's amazing Bethlehem cheese. And then this cheese right here is Bailey Hazen Blue. And so you can see they're both natural rind cheeses. So fungi and the environment colonize those cheeses. This made in Vermont and this made in Connecticut. And so they end up with very different types of microbes on their surface. And so what we're doing in the lab now is, okay, that's cool. What microbes are there? That's great. But why? And how does that impact the flavor of the cheese? So what we're doing is collaborating with chemists, because chemists have really cool tools. And one of the things that they can do is they can actually help us smell the cheese, if you will. What we're doing is using these, so these are tiny little stir bars that are really, really, really small. And what we do is we have them hover over the cheese surface and collect all of the volatiles, all those stinky, wonderful compounds that you enjoy. And what we can do is get really cool chemistry data to link with the microbiome data, the microbes that are present. And also, at the end of the day, it matters not what a GCMS can smell, but what you can smell as a consumer. And so what we're able to do is link microbes, chemistry, and consumers by having people sitting around and talking about how they perceive these cheeses. And this is so cool, and this is super powerful, and this is what keeps me off the streets of Boston and allows me to hang out with wonderful people like Sister Noella and Matteo as well. Yeah, so this is the future of cheese, if you will.
Thank you. Wow, that was amazing. So I think we have a few minutes for questions. So how about I ask all of you to come up here, and you guys can think of things you want to ask in the meantime. Any questions? Okay, how about, and then we have microphones, so if the people in the back with microphones, if you get ready, we have a question up here.
What was my education? I'm a late bloomer. I studied community economic development and really focused on microfinance. I worked in India, Nepal, and Bangladesh, some in Central America, and I'm applying that education to our work really because cheese is a form of capital, and the sellers, it really is a bank, and we're trying to reimagine how we're financing the working landscape through novel ways of providing financial services, ultimately. But I went to a Quaker program at Long Island University, and I started when I was 27. I have a bachelor's degree, and I'm very thirsty for knowledge, which is like why I love hanging around with these people. But I'm a professional amateur, really.
More questions? Is there anyone with a microphone? Yes, please go ahead.
Hi. There's a lot of subliminal effect of smells. You go from flowers, but here's cheese. Now that's very, very acceptable all over the world to remind people of early experiences. I'm getting at perhaps considering somebody to kind of come on as an intern to explore the effect on people that have a bit of dementia, because music has been found to be very powerful. Of the earlier memories, they'll remember and they'll react. Or autism, possibility.
Mm-hmm. Yeah. Interesting.
Anyone else with a microphone? Yes, over there.
What should one or should one not eat the rind?
The age-old question.
Yeah. Did you already eat it?
It is where all the work is. These rinds are soaked in labor. So I always taste the rind. I don't always eat it. Sometimes it adds dimension. Sometimes it detracts. But it is really where all the work is.
It can be bitter, especially in summer, because the Bethlehem cheese, it ages from the outside in, and the fungi are on the outside, and so it would be more ripe along the edges. And sometimes, because of the breakdown of protein, it'll be more bitter. It's not going to hurt you. It's your choice.
Okay, I see a question up there.
Hi there. So I know Sister Noella and Matteo, you guys have more traditional methods of creating cheese, and how important is it to retain that traditional method of maybe using wooden boards or maybe having natural cultures? And I feel like the regulations right now are favoring more industrialized, stringent, consistent products versus this like blossoming artisanal environment. So can you guys talk about that a little bit?
I think we should tell them. We had an experience when we were in Washington a few years ago, and it was the time when the FDA suddenly said you couldn't age cheese on wooden boards. They were threatening all the artisanal cheesemakers. It sort of came out of the blue, but it just so happened that a group of microbiologists and cheesemakers were meeting in Washington at the same time. So Matteo, who's really an activist in all of this, and Dr. Rachel Dutton and Dennis D'Amico, who is in food safety, went marching off to Congressman Welch's office from from Vermont. And lo and behold, the very next day, a group from Congress, it was bipartisan, probably the last bipartisan act we've had, and Congressman Welsh and Paul Ryan, right at the top, Vermont, Wisconsin, and it said to the FDA, since the time of Adam and Eve, cheese has been aged on wooden boards. Now, who knew Adam and Eve were making cheese? But that act of them marching off to the office and concerned, obviously they had come from big cheese states, that worked, and so far, so good. But there are a group, and Matteo is one, and Ben, and Rachel Dutton, and Kathy Donnelly, others, and Dennis D'Amico, they are staying on it with the FDA. And at meetings at the American Cheese Society, they'll have long conferences and dialogues about these things.
We've really had to use hard science to beat them at this game. If it were up to regulators, none of this would be happening. We have science on our side, and that's a powerful tool to beat a federal agency that says that they only listen and make policy decisions based on unpublished science. So we have 20 lineal miles of wooden shelving at the cellars. And so when some mid-level bureaucrat makes a decision like that, it has huge impacts, not just on our business, but on our whole community, right? Our human community and our microbial community as well.
Yeah. Yeah. And in France, they're doing a lot of interesting research, especially scientists with Rebouchon, where they're studying the smear that's on the wooden boards where it's aged, and they're finding anti-listeria compounds within that smear. So that's the kind of science that hopefully is going to help the cheesemakers say, actually, the diversity we're finding in the wood could be safer. But it's important to say that people who make raw milk cheese are not cavalier on the whole in terms of food safety and listeria and E. coli, O157H7. Everyone takes that very seriously because you have to start with clean milk.
And it's the farming practices that actually produce that microbiology, that base microbiology. So by really focusing on the farm side, but the thing about FDA is they don't regulate the farm side, right? That's not, like, the farms aren't part of their jurisdiction. So our entire regulatory framework really isn't built to properly regulate businesses like ours, and that's very complicated for us. We want good regulation. Mm-hmm. We're not anti-regulation. We just want good regulation.
Do you guys pasteurize your milk, and what does that do to cheese?
Where are you? Who is it?
That's God speaking.
We do produce some pasteurized milk cheeses. About 65% of the cheese that we produce is made from raw milk. We buy milk from 2 other farms that we use for those pasteurized cheeses. The milk that we produce is really directed towards the production of Bailey Hazen Blue, Winnemere, and Alpha Tolman. Those all 3 of those are raw milk. The what does it do part? Well, it wipes everything out. You start with a blank slate, and then you have to try and recreate artificially this diversity. We pasteurize soft cheeses because of the 60-day aging rule, and we found that consumers consistently have a better experience with cheese that's at peak, around 70 days, and there isn't enough a time frame there in the cold chain and distribution to get raw milk cheeses to customers in the best shape. So, yeah.
Why is it so difficult to recreate or emulate terroir? Or is it possible, or if so, is it possible to emulate terroir? That's my question.
Well, I think what was surprising to us is that, for instance, this cheese, the Saint-Nectaire, is from the Auvergne, France, and then suddenly the same fungi are growing on our cheese in Connecticut. So it kind of seems against terroir, but I think in a very important part is the technology in the cheese making. So the techniques that the cheesemakers used for centuries selected for the microorganisms that gave the best cheeses. So salting is really important because, like Geotricum, salt sensitive, Mucor less so. Penicillium can tolerate the most salt. That's why blue cheeses taste salty. And so that's what the cheesemaker manipulates. And then the materials they use, you see this straw mats that it's aged on. That encourages certain fungi to grow. The humidity of the cave, the wood. So I think the cheesemaker and the affineur have to adjust conditions and materials to produce cheeses that they think make the best cheeses. That's also selecting for the best cheese ripening microorganisms. And certainly if you're using native strains, that's going to, I think, increase this taste of place, which is what Matteo refers to as terroir. That's the American version of terroir.
Can I take a stab at it too?
Yeah.
So we manage our farms to produce specific cheeses, right? And it's like mostly Bailey Hazen Blue and Windermere. So these are kind of higher risk category cheeses. We're actually looking for lower total bacteria counts. For alpha Tolman, and we're going to be building a farm specifically to produce alpha Tolman, a hard cheese. What we're looking for in a hard cheese is higher total bacteria counts. So in the Jura and the production of Comté, certain times of the year, they only wash their equipment once a day. The equipment becomes a vector where they're actually incubating microbes. So in the evening, it's actually in the morning, they'll flush their milking system with scorching hot water, which is selecting for thermophilic microbes, and they allow those thermophilic microbes to colonize their milking system, and then they milk through that milking system and collect all those microbes in the evening to try and maximize the total number of bacteria. They also don't use teat dips, no chlorine sanitizers anywhere on the farms or in the dairies. They use wood wool, so this wood wool to clean the udders. They don't use teat dips or sanitizers. They're trying to preserve. 80% of the microbes in raw milk come from the outside of the udder, right? So for a hard cheese, you know, and they know this in the Jura. INRA at Poligny has done a huge amount of research, and the biggest threat to the quality of Comté cheese is sanitation and the decline of microbial populations in raw milk, right? So if you're making a hard cheese, you're going to have a completely different set of practices than if you're making a soft cheese. And that's why you can't just have one answer to that. So how do you recreate that? That kind of diversity it would be very, very difficult because the range of microbes that are growing in the richness of that microbial ecology is impossible to recreate. It's like the sum of the practices in a farming system, right? And you know, if you're making pasteurized cheese, you might be able to, you know, add 10, 15 different strains of different cultures. And so you can get part of the way there, and you can get some of the important ones there, but terroir is a much bigger idea than just, the microbes. It really is like a system, just like how you farm. Yeah.
Okay, so let's end here, but please feel free to stay around for a while. And thank you again to Sister Noella, Matteo, and Ben.